we begin with the event, which is the Explorer series, let me take a minute to quickly introduce our organization, which is the Welcome Trust TBT India Alliance. So the school children might definitely haven't heard of the organization. What we do is, since our establishment 10 years ago, we fund scientific research in the country, in India. And this scientific research is primarily aimed at improving human and animal health. Apart from funding scientific research, we also love coming to you people, to the public, to share the science and the excitement of science with the public at large.
तो आज के इवेंट का नाम हमने एक्सप्लोरर सीरीज रखा है। इसका नाम एक्सप्लोरर सीरीज ही क्यों रखा है हमने? तो बेसिकली एक्सप्लोरर शब्द का मतलब होता है, एक्सप्लोरर शब्द एक्सप्लोर से आता है बेसिकली, और एक्सप्लोर का मतलब होता है खोज करना। एक साइंटिस्ट का काम होता है खोज करना, फिर चाहे वो स्पेस में करे, लैब में करे, माइक्रो ऑर्गेनिजम्स के साथ करे, या फिर एक क्लिनिक में करे।
तो ऐसे ही 4 एक्सप्लोरर्स आज हमारे साथ हैं। मैं क्विकली जो हमारे फर्स्ट एक्सप्लोरर हैं उनके बारे में आपको बताऊंगी। तो फर्स्ट है खुद हमारे शंकर। शंकर इज अ माइक्रोबायोलॉजिस्ट, ही स्टडीज माइक्रो ऑर्गेनिजम्स। He has a master's degree in genomics and PhD in microbiology from Madurai Kamaraj University in Tamil Nadu. After his PhD, he received postdoctoral training for 3 years at the University of Kansas Medical Center in USA. After that, he studied at the universities. He studied how bacteria cause tooth decay, which a lot of us face. And now he's an assistant professor here at IIT Jodhpur. He's also an India Alliance fellow. He enjoys teaching, and a fun fact about Shankar, which a lot of you would relate with, is he's a huge Harry Potter fan, and he loves using those quotes to apply in his real life situations. Please welcome Shankar on the stage. Very important announcement for all of you. We've already told you to please be interactive and ask questions to the speakers after their talks. And we have prizes for you. So if you ask questions, we'll pick the best ones of them, and you'll get prizes, exciting prizes for you. So please try to be interactive.
I'd like to thank ITJ and India Alliance for making this happen. I am Shankar Manoharan. Sorry, thank you so much for that wonderful introduction and the fun fact. I'm pretty sure none of my students here know that about me. I'm also an India Alliance Early Career Fellow and an assistant professor at IIT Jodhpur. I joined the institute in 2017 and have been here since. Today I'll be taking you through my journey where I fell in love with bacteria, the good, bad, as well as the ugly ones. But have you wondered how something so small that you guys can't even see it has somehow the power to bring down a fully grown human being with disease? Right? This is the fascination that I grew up with. My parents are both doctors, and every time they talk to me about a patient, I always used to wonder, how is this that something so small can actually cause such severe disease in a fully grown human being? So to introduce a bacteria, this is a typical textbook drawing. It looks something like this, and the long tails that you're seeing over here, these are flagella, which the bacteria uses to move around. They rotate, and it actually propels the bacteria around. Then you have a nice cell envelope, you have the cytoplasm, you have the DNA inside. This is a textbook representation, and this is just an electron micrograph of a bacterial cell. Right? So let's briefly go over why we should study bacteria, because they are so minute that we should probably not care about them, right? Do you agree? Should we care about them or should we not? Okay, so I'd love for this session to be very interactive. You guys don't have to be very shy in communicating. So the interesting fact is that bacteria are everywhere. Even though you might not see them, you're interacting with them every day, you're eating them every day, You're shedding them every day in your skin, everywhere, right? So typically in a plant, if you see, each area contains a distinct microbial population. The leaves, the roots, the shoots, each part of a plant. Same thing goes for a human being. Each part of the body has a distinct microbial population. Bacteria in your ears might not necessarily be the same as the bacteria in your gut. And because each environment is different, it's very important for us to understand who we are living with, right? Like your roommates. If you want to know more about your roommates, it's helpful, right? So, and you're going to live with these bacteria for the rest of your life. Having said that, what caused my interest in microbiology or bacteria in general? So I was just a college kid back in 2005 when this Nobel Prize in Physiology and Medicine came out. Dr. Barry Marshall and Dr. Robin Warren. They got awarded for the discovery of a bacterium. The name is Helicobacter pylori, a bug which was completely newly discovered, right? Okay, so let's do a quick pop quiz. What is an ulcer, a gastric ulcer? How do you get an ulcer? You get an ulcer because of stress or because of not having good eating habits, not having regular timings for your meals, right? Unfortunately, that's not true. There's a bug that can cause ulcers as well, and that's Helicobacter pylori, and that's why these two got the Nobel Prize. So my first fascination with Helicobacter pylori was because it was the perfect predator. It had everything that is required to actually cause a gastric ulcer. Just think about it. The pH of the human stomach is so acidic that it digests all the food, right? How can a bug live there? How can a bacteria live there? It's one of the harshest environments known to man. And still, this guy has the ability, almost perfectly engineered ability, to survive in this environment. Not just survive, but to multiply and cause disease. If you look at it quickly, it has this flagella that I showed you a little bit before, that it can use to tunnel through the mucus layer that's present in the gastric environment, reach the cells, and then it injects the cells with certain effectors and causes these ulcers. Beautiful, isn't it? So this is where I got drawn to molecular microbiology, where I started looking inside bacterial cells, how they function and what they do. Now, once this curiosity was triggered, I decided to take up science as a career, and I went on to do my PhD at Madurai Kamaraj University. Science and life are never fair. I was thrown into a completely different project. I ended up going into plant-microbe interaction. And that's the first segment of my talk, where I'll be talking about the good bacteria, right? So, plant-microbe interactions, referring to the bacteria which live in association with plants. They do quite a lot of functions. They they fix atmospheric nitrogen, they solubilize nutrients, they synthesize certain phytohormones. And as I've shown here, when you treat plants with certain strains of bacteria, they tend to grow better because of all these benefits, right? But to maximize this benefit, it's necessary for the bacteria to live in close association with the plant. And that is achieved by colonization of the plant. Colonization is nothing but living in association with something. So there are certain microbes which have colonized you. And similarly, if a microbe can colonize a plant, the picture is not very clear, but what you will see is there are single cells. The bacteria are shown in green here. Single cells attaching to the root surface, and then these cells are slowly growing into a colony, a microcolony, a cluster of cells, right? And then towards the end, at the end of 7 days, you'll see nice thick patches of aggregates. So what you are essentially seeing is that single cells colonizing on a surface and then multiplying to become multicellular aggregates, right? Now, what would you do in a classroom if there are just 2 of you there? You would probably talk to each other, right? If the number increases to 20, the sound increases correspondingly. And then when it becomes 50, it becomes a fish market. correct? Same thing happens with bacteria as well. When many cells live together, they start talking to each other. And I was interested at that point to find out how are they doing this? How are they talking to each other? And that's what sparked an interest. And we figured that they do talk to each other, and they talk to each other through signals. Not electrical signals, but chemical signals. They produce certain molecules which the other bacteria can sense. And this phenomenon is called quorum sensing. Right? So to put it simply, it looks something like this. Some bacteria have an antenna-like structure. So the antenna is just a metaphor here. It's a signal transmitter, and then you have a dish that receives the signal, right? So the bacteria from here can communicate to the other bacteria and say, hey, come, let's go colonize that plant. Then roger that, we'll move to the plant root as soon as possible, right? So both need to have both the transmitter as well as the receiver for this to work, right? But in the bacteria that I was working on, the one that I just showed you before, there was a surprising twist. We saw that there was no antenna, no mechanism to transmit signals, but the bacteria had the receptor. It had the receiver dish. That was weird. Why would a bug have a receptor for a signal that it cannot synthesize? Right? And we started wondering, maybe they're not just communicating probably, but if they're not communicating, then why do they need to have a dish, a receiver? And at this point, we thought a little out of the box. Then we figured that the plant was doing the talking. The plant was sending out signals, which the bacteria was receiving, right? And then the plant was actually telling the bacteria that I have sufficient number of bacteria living with me, so go find a different place and live. Right? So the signals were actually discouraging the bacteria from coming close to the plant. So this was one of the modules where I just spoke about the good bacteria, the ones that benefit plants. Now we'll move on to the bad one, which is, so now we'll have to go back to the University of Kansas Medical Center where I did my postdoctoral training. As she rightly said, that's where I did some work on tooth decay, which is a problem that all of us live with. So introducing Streptococcus Mutans. Now this guy, if you look at this micrograph over here, you will see that there is a kind of a scaffold set up, and bacterial cells are living within it, right? This is a clear example of a biofilm, bacterial community living together, right? And guess where this is? It's on your teeth right now, if you haven't brushed in the morning, right? So this is classic dental plaque, which, when magnified, looks like this. And one of the major components of this plaque is actually Streptococcus mutans, which is also the causative agent of your dental caries. So I'm sure most of you would have heard from your elders not to have too much sweets before bed. And it's bad for your teeth for good reason. Why? Because the sucrose that you eat, sucrose is the sugar, right? And the sucrose that you eat is taken in by this bug and is converted into glucan. Glucan is a polysaccharide. It's a polymer, something like ferricol, which helps the bug stick to other bugs as well as stick to the tooth surface over here, right? So if you take sugar, what happens is it gets converted into a paste-like material which helps the bacteria to stick tightly to surfaces, right? And when this happens, in the subsequent step, the same sugars get converted through different metabolic pathways into acids. Now, all of you must have almost seen this video where they drop a tooth into a bottle of Coke and it gradually dissolves, right? Same thing happens in a smaller scale in your tooth when these acids get released onto your tooth surface, resulting in dental caries, which are extremely painful, right? So during my stay at the University of Kansas Medical Center, we unraveled certain mechanisms of gene expression which control this process. and we identified a protein which we could effectively use to target this bacterium from ever attaching to the tooth. Right? So this is about one of the bad bacteria that I've dealt with. And moving on, I want to talk to you about the worst one that I've encountered so far, the ugly bacteria, which essentially kills the weak, right? So you would call such a person cruel, right? If somebody kills a weak, innocent thing. So this is what the hospital-associated pathogens do. So why do I call it hospital-associated? Normally you go to the hospital to get yourself treated for an infection. But today, if you go to a hospital, it's likely that you will catch an infection there, provided your immune system is not functioning fully, right? So if your immune system is not functioning fully, your defenses are on the low, and there is a group of bacteria called the escape group, the names of which I've just listed out here. They will almost certainly infect and will cause subsequent infection, right? Now, you might ask me, what if I catch a bacterial infection? We still do have antibiotics, right? Why don't I just take antibiotics? Problem is, we live in an age where antibiotics no longer work. Can anybody tell me why? Antimicrobial resistance, right? So why do antibiotics not work? So in any healthy environment, you will always see that there is a mix of antibiotic sensitive as well as antibiotic resistant bacteria. Fine, let's do another pop quiz. Those of you who take antibiotics for a common cold, please put up your hands. How many of you have been prescribed antibiotics for a cold? Your doctor gives you antibiotics for a cold. And moving on, I want to talk to you about the worst one that I've encountered so far, the ugly bacteria, which essentially kills the weak, right? So you would call such a person cruel, right? If somebody kills a weak, innocent thing. So this is what the hospital-associated pathogens do. So why do I call it hospital-associated? Normally you go to the hospital to get yourself treated for an infection. But today, if you go to a hospital, it's likely that you will catch an infection there, provided your immune system is not functioning fully, right? So if your immune system is not functioning fully, your defenses are on the low, and there is a group of bacteria called the escape group, the names of which I've just listed out here. They will almost certainly infect and will cause subsequent infection, right? Now, you might ask me, what if I catch a bacterial infection? We still do have antibiotics, right? Why don't I just take antibiotics? Problem is, we live in an age where antibiotics no longer work. Can anybody tell me why? Antimicrobial resistance, right? So why do antibiotics not work? So in any healthy environment, you will always see that there is a mix of antibiotic sensitive as well as antibiotic resistant bacteria. Fine, let's do another pop quiz. Those of you who take antibiotics for a common cold, please put up your hands. How many of you have been prescribed antibiotics for a cold? Your doctor gives you antibiotics for a cold. Cold is a viral disease, and antibiotics do not work on viruses, right? So every time you're taking antibiotics for a common cold, what you're essentially doing is you're killing off your normal microbiota while letting the antibiotic resistant ones survive. And this will eventually lead to multiplication of the antibiotic resistant bugs, which will, if given the chance, infect you to cause disease. And then when you go for the actual antibiotic, the antibiotic doesn't work anymore because they're all resistant to the antibiotic, right? And it doesn't stop there. It actually, they also spread the antibiotic resistance among antibiotic sensitive bacteria, ultimately leading to more chaos, right? So this is why antibiotics don't work. So in fact, it's not just restricted to non-prescribed antibiotics, but it is also that
our foods, the meats that you eat, the milk, the vegetables that you eat are all laced with antibiotics because the food industry now uses antibiotics to promote growth, right? And all this is ending up in the food chain, ultimately coming to bite us humans. So even without prescription antibiotics, many of us here will be taking some antibiotics without our knowledge. And this altogether fuels this phenomenon called antibiotic resistance, where we are running out of usable antibiotics. And this is where we come into the picture. So we want to develop technologies where we want to look at other methods, non-antibiotic methods to subdue bacterial infections. So let me talk a little bit about the bug that I work with. It's Klebsiella pneumoniae. It's a hospital-associated pathogen. So this guy, the very characteristic of this bug is that it produces a very thick capsule layer, right? So the purple ones in the middle are the bacteria, and the white surrounding is the capsule. So the capsule is extremely important for the bug. And in the absence of capsule, the bug will be almost always killed by our own immune system, right? So if this layer, the capsule that surrounds the bacteria, is eliminated, the bug becomes a softer target for us to kill, right? And that is the kind of direction in which we are working in right now. We are trying to understand how Klebsiella synthesizes its capsule, and if we can suppress the synthesis of its capsule. Now, for those of you where capsule kind of seems too foreign, think of a force field, right? All of us know what a force field is. If you are inside the force field, it's not going to hurt you. Any attack is not going to hurt you, right? So the capsule is like a force field for bacteria. So when the capsule is on, no attack from the human immune system is going to harm the bacteria in any way. So the bacteria is still alive, right? But in case the capsule gets turned off, the human immune system can step in and kill the bug. So again, to easily visualize, if you remove the capsule, our immune system can sense the bug and effectively destroy it, right? So my lab here at IIT Jodhpur is working towards disabling the capsule of Klebsiella pneumoniae. We are trying to understand how Klebsiella synthesizes its capsule, the pathways that are involved, the regulatory control mechanisms that are in place, and then we'll try to target them with specific drugs or small molecules to effectively suppress the synthesis of capsule. So with that, I'd like to conclude, and there are some key takeaways from here. We share the world with bacteria, and they live with us, performing key functions. It's also important to understand that we should not jump onto the bandwagon and say all bacteria are bad. There are some good ones as well. And some bacteria beneficial to human beings in various contexts, whereas some are harmful to us. And we should learn more about bacteria as we go ahead, so that in case we need to fight them, we should be prepared to fight them. Because you need to understand your enemy better to kill him or her, right? So understand your enemy as well as understand your friend. So that's why we do what we do here at IIT Jodhpur at the Microbial Physiology Lab. I'll be happy to take any questions. I request that you please mention your name as well as the school, college or institution that you are from. Thank you.
You said that some bacteria develop antibiotic resistance. How can they do this?
Resistance when they are constantly exposed to antibiotics, right? So if we are inadvertently exposed to antibiotics a lot of times, our microbiota will develop resistance slowly over time. All bacteria do have the ability to develop resistance, but that doesn't mean that all of them do.
So I am Gargi Verma from school DPS Sali, and I want to ask that if अगर प्लांट भगाते भी हैं बैक्टीरिया उनके केमिकल सिग्नल भेज के तो बुलाते भी हैं क्या?
Yes. So see, in my case, the plant that I was using was rice, and the bacteria was Helicobacter. So maybe rice didn't want too much of Enterobacter to live with it. That's why it said no. But what normally happens is the plants secrete certain substances through their roots, and these are called root exudates. And these exudates actually contain a lot of sugars, amino acids, nutrients, and that attracts all the bacteria to us. Right? And there might also be chemical signals which can attract bacteria towards plant roots, specifically where we talk about this nodulating plants, right? Where root nodules are there, bacteria live within them. So bacteria have to be attracted to the root, and then they have to get inside the root to form nodules. These are achieved by chemical signals.
Sir, I want to ask that you have said antibiotics does not work on like viruses, like common cold. But I heard somewhere that small amount of alcohol works in it. Sir, is there any logical?
Does kill microbes. Right? So alcohol will, if used at the right percentage, can kill microbes. But it doesn't mean that you have to consume alcohol to actually kill microbes. It doesn't work that way. So alcohol is used as a surface still, where we wipe surfaces with 70% ethanol to just kill off bacteria.
So my question is this, that we are suffering from common cold, so antibiotics doesn't work on viruses. So then why doctor prescribe us to take antibiotics?
It's done to prevent secondary bacterial infections. So when you're when you're suffering from a common cold, your immune system is already fighting the cold, right? So it may be to actually prevent secondary bacterial infections, which may co-occur. This is my understanding. If Dr. Kapoor has a better response.
Thank you, sir.
For this excellent talk.